Hello friends, I am Dr. Neelam Mahajan. I shall be discussing parenteral products part 1 formulation today. In this module, we will have an introduction to parenterals. Then we will talk about requirements of parenterals. We will also learn about factors affecting formulation of parenterals and formulation development including formula ingredients. Parenteral products are one of the unique sterile dosage forms of therapeutic agents free of viable microorganisms and are injected through the skin or mucous membranes into internal body compartments. Parenteral preparations may be given by various routes including intravenous, intramuscular, subcutaneous, intradermal and intraperitoneal. Via an intravascular route, complete drug availability occurs immediately and no absorption is necessary. For all other routes, at least a blood vessel wall and usually one or more tissue cell walls must be permeated before the drug can enter the circulation. Most often, this occurs by passive diffusion and is most favorable when drug has both lipophilic and hydrophilic properties, with the former being predominant. With non-vascular routes, absorption is affected by factors which include the size and number of blood vessels supplying the tissue, the movement of the tissue following injection, the physical and chemical properties of the drug, and such characteristics of the dosage form as to whether it is a solution, suspension or emulsion, and the nature of the vehicle and its pH. Once in the circulating blood, the physiological effect of a therapeutic agent is affected by the extent to which it distributes throughout the body, by the degree of binding to plasma proteins, and by its rate of elimination by hepatic metabolism or renal excretion. Intravenous and intraspinal preparations are rarely given in a form other than aqueous solutions. The danger of blockage of fine capillaries, particularly in the brain, precludes the use of forms other than solutions for IV administration, although emulsions with carefully controlled particle size of the dispersed phase are reported. For intraspinal medication, the sensitivity of the nerve tissues generally precludes the use of anything but the purest of solutions. Preparations given intramuscularly, subcutaneously or intradermally can be given as solutions, suspensions or emulsions. Even solid pellets may be implanted subcutaneously or intramuscularly. The vehicles can range from water for injection to glycols to fixed oils. Although care must be taken to avoid undue tissue irritation, mild local irritation is permissible at these injection sites. Now the requirements of parenteral products. Since parenterals evade the highly efficient first line of body defense, that is the skin and the mucous membranes, they must be free from microbial contamination and toxic components, should possess an exceptionally high level of purity, and all components and processes involved in their preparation must be selected and designed to eliminate contamination of all types as far as possible. The nature of a preparation can influence significantly onset and duration of therapeutic effect of a drug and the form of the absorption pattern achieved. Therefore, there is a need to develop the formulation for a parenteral by its careful integration with its intended administration in a patient. This further requires that the physical and chemical properties of a drug must be determined and the interaction of the drug with any desired excipients must be studied, and the effect of each step of the process on its stability must be studied and understood. Now let's have an understanding of the factors affecting formulation of parenterals. The first factor affecting the formulation of parenterals is the route of administration. The intended route of administration has a marked effect on the formulation of a parenteral. In general, only solutions of drugs in water may be given intravenously. Suspensions may not be given because of the danger of blockage of the small blood vessels. Aqueous or oleaginous suspensions and oleaginous solutions cannot normally be given subcutaneously because of the pain and irritation caused. Muscle tissue tolerates oils and suspended particles fairly well, and is therefore the only route normally suitable for their administration. The administration of a drug deep into muscle tissue results in a pool of the product at the site of injection. From this depot, the drug is released at a rate determined to a large extent by the characteristics of the formulation. Whether the solvent is aqueous or oleaginous affects the rate of absorption. Oleaginous solutions are usually more slowly absorbed. Increasing the viscosity of solutions slows the absorption, just like gelatin or polyvinyl pyrrolidone in water and aluminum monostearate in oil. Utilizing modifications of the drug molecule to render it less soluble permits the production of stable suspensions, causing a marked reduction in the rate of absorption of the drug from the depot. Thus, utilizing various modifications in the formulation of the product makes it possible to retard the rate at which a drug is released from a depot. The second factor is the volume to be administered. Volume in which a dose of the drug must be included is the second factor affecting formulation of parenterals. For intracutaneous injections, a volume of more than 0.2 ml is rarely used because tissue volume is small and compact and slow absorption takes place due to the lack of blood vessels. Volume of 1 ml or less may be injected subcutaneously, and only occasionally are volumes of more than 2 ml are given intramuscularly. Volumes of 10 ml or less may be given intraspinally, but large volumes may be given safely only by the IV route with carefully controlled rate of administration. It is not easy to administer a volume greater than 20 ml by a syringe and impractical to set up an infusion unit for less than 250 ml. Isotonicity is the third factor affecting the formulation of parenterals. It is a characteristic that is preferable for the comfort of the patient. It has greatest importance for intraspinal injections because the circulation of the cerebrospinal fluid is slow and disturbances of osmotic pressure quickly cause headache and vomiting. Since intracutaneous injections are given mostly for diagnostic purposes, non-isotonic solutions may cause false signs of irritation. Isotonicity is not essential for subcutaneous injections. For drugs given intramuscularly, a slightly hypertonic solution may increase the rate of absorption of drugs by causing local effusion of tissue fluids. Now we will discuss the type of parenteral products. First type of products are freeze-dried products. Solutions intended to be freeze-dried must be aqueous because drying involves the removal of water by sublimation. Furthermore, stability problems related to aqueous system are practically non-existent due to the existence of solution for only a brief period during processing. However, the formulation must reflect the characteristics of a solid residue after drying and solution after reconstitution at the time of use. Often the drug alone does not give sufficient solid residue or characteristics appropriate for the product, including a uniform color and texture, a supporting matrix of solids sufficient to maintain essentially the original volume after drying, and sufficient strength to prevent crumbling during storage. Therefore, substances are added to provide these characteristics. In addition, the nature and amount of solids in the solution largely determine the eutectic temperature of the frozen solution, the sub-zero temperature at which the frozen material will melt, which determines the temperature below which the product must be held during freeze drying. It also determines the rate of thermal and vapor transfer through the product during the process of drying and rate of solution of the product during reconstitution. The percentage of solids in the frozen plug should be between approximately 2 to 25%. Best salts for providing uniform crystal size, uniform color and texture, physical strength, and rapid reconstitution include monobasic and dibasic sodium phosphates. Sodium chloride is often used, but when used alone, cake tends to shrink remarkably in volume and appears crusty and crumbly. When organic substances are used to provide solids for the cake. Care must be taken during heating, particularly during terminal stages of drying, to avoid discoloration of cake by charring. Added substances required in the formulation must not be volatile under conditions of drying. Therefore, antibacterial agents like phenol and benzyl alcohol should not be used. After discussing the freeze-dried products, now let's talk about long-acting formulations. Long-acting parenteral drug formulations are designed ideally to provide slow, constant and sustained release of a drug over a prolonged period of time, essentially to stimulate and replace the more hazardous continuous intravenous infusion of a drug. Though ideality is difficult to achieve, yet depot dosage forms that approach the desired goal are being developed and include dissolution-controlled depot formulations, depot formulation produced by binding of drug molecules to adsorbents, encapsulation type depot preparations, and esterification type depot preparations. In dissolution-controlled depot formulation, the rate of drug absorption is controlled by slow dissolution of drug particles with subsequent release to tissue fluid surrounding the bolus of product in the tissue. Formation of drug salts with very low aqueous solubility is one of the most common approaches to this type of formulation. Control of particle size also can contribute to slow dissolution. Further, the suspension of the drug particles in vegetable oils, and especially if dealt with substances like aluminum monostearate, produces prolonged absorption rates. Second type of depot formulation is produced by binding of drug molecules to adsorbents. Only the free portion in equilibrium with that which is bound can be absorbed. As drug is absorbed, a shift in equilibrium is established and drug is slowly released from bound state to free state. This is particularly exemplified by binding of vaccines to aluminum hydroxide gel to provide a sustained release. A third type of depot preparation is encapsulation type, in which biodegradable or bioabsorbable macromolecules like gelatin, phospholipids and long-chain fatty acids become a diffusion matrix for the drug. Drug is encapsulated within the matrix and release of drug molecules is controlled by a rate of permeation out of diffusion barrier and by rate of biodegradation of the barrier macromolecules. A fourth type of depot preparation is esterification type, in which esters of a drug that are bioerodible are synthesized. Esterified drug is deposited in tissue at the site of injection to form a drug reservoir. The rate of drug absorption is controlled by partitioning of drug esters from reservoir to tissue fluid and by the rate at which the drug ester regenerates active drug molecules. Very often these esters are dissolved or suspended in oleaginous vehicles which further slow the release. Now the third type of products, suspensions. Parenteral suspensions have 0.5 and 5% solids content but may range up to 30% in some antibiotic preparations. Amount of solids and nature of vehicle determines the viscosity of the product as well as its syringeability. The most important requirement for parenteral suspensions is a small and uniform particle size. Small uniform particles lead to slow uniform sedimentation rates and predictable dissolution and drug release rates. Small uniform particles lead to a reduction in the tendency for larger crystal growth during storage and easy syringeability. Suspension stabilization for the period between manufacture and use is challenging. Solids gradually settle and may cake, causing difficulty in redispersion prior to use. Surface active agents may help in the preparation and stabilization of a suspension by reducing the interfacial tension between particles and vehicle. Polysorbate 80, lecithin among the surface active agents used in parenteral suspensions. Concurrent addition of a hydrocolloid like sodium carboxymethylcellulose may enhance the effect of surfactant and cause loss of surface charge of dispersed particles, water repellency and tendency to agglomerate. Occasionally, parenteral suspensions may be improved by a slight increase in viscosity, either by increasing the amount of protective colloid or by adding a compound such as sorbitol. In other formulations, flocculation of suspended particles is necessary to prevent packing to a dense cake. Addition of selected ions that increase the surface charge of the solid particles may cause them to form fluffy aggregates. These settle rapidly but to a large sedimentation volume which can easily be redispersed. Monosodium citrate has been used effectively for such a purpose. Now talking about emulsions. Preparation of a parenteral emulsion is troublesome. It is made more difficult by rigid requirement for particle size control to prevent embolus in blood vessels, by the limited choice of emulsifiers and stabilizers of low toxicity, and by the preservation of the oil phase against development of rancidity. Principal problem in the formulation of parenteral emulsions is the attainment and maintenance of uniform oil droplets of 1 to 5 micrometers in size as the internal phase. In emulsions, difference in density between oil and water is relatively small, hence separation of phase does not occur as readily as with suspensions. Dispersed phase should have droplet size of less than 1 micrometer, and emulsion must be stable to autoclaving. Elevated temperatures, however, tend to produce coalescence of the dispersed phase, and excessive shaking can cause acceleration of the rate of creaming. After having an understanding of the type of parenteral products, now let us discuss the formulation development and formula ingredients. The final objective in the development of a sterile product is elicitation of a therapeutic effect in a patient. Formulation of a sterile product involves combining one or more ingredients with a medicinal agent to enhance the convenience, acceptability, or effectiveness of the product. Rarely is it preferable to dispense a drug single as a sterile dry powder unless the formulation of a stable liquid preparation is not possible. The first formula ingredient is therapeutic agent. A therapeutic agent is a chemical compound subject to physical and chemical reactions characteristic of the class of compounds to which it belongs. Therefore, a careful evaluation must be made of every combination of two or more ingredients to ascertain whether or not adverse interactions occur, and if they do, of ways to modify formulation so that reactions are eliminated or minimized. Information concerning basic properties must be obtained before an intelligent approach to formulation can begin. Important properties of a drug or of the total formulation are evident only after product has been stored or used for a prolonged time. The second formula ingredient is vehicle or solvent system. Under this, we will discuss aqueous systems first. By far, the most frequently employed vehicle for sterile products is water, because it is the vehicle for all natural body fluids. One of the most inclusive tests for quality of water is the total solids content, a gravimetric evaluation of the dissociated and undissociated organic and inorganic substances present in water. However, a less time consuming test, the electrolytic measurement of conductivity of water, is the one which is most frequently used. The validity of measurement as an indication of purity of water is inferential, in that methods of producing high purity water like distillation and reverse osmosis remove undissociated substances along with those that are dissociated. Undissociated substances like pyrogens, however, could be present in the absence of ions and not be disclosed by the test. Therefore, for contaminants other than ions, additional tests should be performed. Additional tests for quality of water for injection with permitted limits are described in USP monographs. When comparing total solids permitted for water for injection with that for sterile water for injection, considerably higher values are permitted for sterile water for injection, since it has been sterilized usually by a thermal method in a container that had dissolved to some extent in the water. Therefore, the solids content will be greater than for the non-sterilized product. On the other hand, The 10 ppm total solids officially permitted for water for injection may be too much high when used as vehicle for many products. Water shall contain a minimal amount of organic compounds which are undesirable for two main reasons. They may be toxic and may serve as source of nutrition for microorganisms. In practice, water for injection normally should have total organic carbon not more than 500 ppm. Now the non-aqueous and mixed solvents. In formulation of sterile pharmaceutical products, sometimes water is eliminated entirely or in part from the vehicle primarily because of solubility factors or hydrolytic reactions. A non-aqueous solvent must not be irritating, toxic or sensitizing and must not exert an adverse effect on the ingredients of the formulation. Screening of such a solvent must include an evaluation of its physical properties. Solvents that are miscible with water and solvents that are usually used in combination with water as vehicle include propylene glycol, glycerin and ethyl alcohol. Water immiscible solvents include fixed oils, isopropyl myristate and benzyl benzoate. Most frequently used non-aqueous solvents are polyethylene glycol, propylene glycol and fixed oils. Now we will talk about solvent selection when a parenteral therapeutic agent is preferred as a solution. The solutions are preferred because aqueous solution is physiologically compatible with body tissues and biological response elicited is reasonably predictable. High dielectric constant of water makes it possible to dissolve ionizable electrolytes and its hydrogen bonding potential dissolves organic substances. Conversely, water is a poor solvent for non-polar compounds. Since therapeutically active compounds given by injection range in property from highly polar to non-polar, solvents having complementary properties must be used if a solution is to be achieved. Solvent selection requires that solvents to be injected must have low toxicity to body tissues. Ether is a solvent for testosterone but is highly irritating to body tissues and cannot be used as a solvent alone for an injectable preparation. Frequently, the desired solubility can be achieved with mixed solvents, for example, use of approximately 40% ethanol in water to solubilize digital glycosides. Compounds that are dissolved in water are often subject to degradative reactions. Formulation must be designed in such cases to minimize these degradative effects. Often these reactions are markedly affected by the pH of the solution. The use of a mixed solvent system often reduces degradative reactions. Oleaginous injections are, however, viscous and involve frequent incidence of pain upon injection. After learning about solvents, now we will discuss solutes. Physical and chemical purity of solutes used for sterile preparations must be exceptional. For a few substances, special parenteral grades are commercially available. In addition, solutes should be free from microbial and pyrogenic contamination. Not only proper quality of chemical should be procured, but also storage conditions should prevent contamination, particularly after a container has been opened. Preferably, production lots should be designed to use the entire contents of packages of chemicals wherever possible. Added substances are the next formula ingredients. Substances added to a product to enhance its stability are essential for almost every product. But these agents must not adversely affect the product and must be non-toxic in the quantity administered to the patient. They should not interfere with the therapeutic efficacy or the assay of the active therapeutic compound. They must also be present and active when needed throughout the useful life of the product. Therefore, these agents must be selected with great care and must be evaluated as to their effect upon the entire formulation. Such substances include solubilizers, antioxidants, chelating agents, buffers, etc. and numerous other substances for specialized purposes. Now the antibacterial agents. Antibacterial agents in bacteriostatic concentration must be included in the formulation of products packaged in multiple dose vials and in formulations to be sterilized by marginal processes or made by aseptic manipulation. Then antioxidants. Antioxidants protect a therapeutic agent susceptible to oxidation, particularly under accelerated conditions of thermal sterilization, and may function in at least two ways. That is, by being preferentially oxidized and by blocking an oxidative chain reaction in which they are not usually consumed. In addition, certain compounds act as synergists and increase the effectiveness of antioxidants. Certain compounds complex with catalysts accelerating the oxidative reaction. For those products in which oxygen enters into a degradative reaction, an antioxidant effect can be achieved by displacing oxygen from contact with the product. Next formula ingredients are buffers. These are added to maintain the required pH for many products, as change in pH may cause significant alterations in the rate of degradative reactions. Buffers must have the capacity to maintain pH of the product, but not enough to prevent body fluids from overpowering the buffer following administration. In most cases, biological effectiveness of the drug is maximum at or near the biological fluid pH rather than at the stabilizing pH of the injected product. Acetates, citrates and phosphates are principal buffer systems used. Buffer systems must be selected with consideration of their effective range, concentration and chemical effect on the total product. Now tonicity contributors. Compounds contributing to the isotonicity of a sterile product reduce pain of injection in areas with nerve endings. Simple electrolytes and non-electrolytes like glycerin and lactose are most commonly used for this purpose. These are usually the last ingredients added to the formulation after other ingredients in the formulation are established and osmolality of the formulation has been measured. Isotonicity actually depends on the permeability of a living semi-permeable membrane separating the solution from a biological cell system. Most frequently, for sterile pharmaceutical preparations, membrane concerned is the one enclosing the red blood cells. Therefore, a preparation cannot be considered to be isotonic until it has been tested in a biological system. Chelating agents are the next formula ingredients. They are non-ionizable trace amounts of heavy metals, catalyze degradative changes. Chelating agents most commonly used to tackle this problem include trisodium or calcium disodium salt of ethylene diamine tetraacetic acid in a concentration of about 0.05% W by V. Heavy metals extracted from rubber closures also may be bound by a chelating agent, reducing the possibility of reactions with ingredients in the formulation. Next, we will discuss inert gases. These have been used to displace oxygen from a solution and to reduce the possibility of oxidative changes in the formulation. Inert gases may be used to stabilize solutions in other ways. For example, sodium bicarbonate injection decomposes, particularly during autoclaving, to produce sodium carbonate, carbon dioxide and water. Saturation of solution with carbon dioxide inhibits this reaction and stabilizes solution. Lastly, we will discuss protein stabilizers. A number of ingredients have been shown to stabilize proteins both in dry and solution state. Serum albumin competes with therapeutic proteins for binding sites in glass and other surfaces and minimizes the loss of protein caused by surface binding. Cryoprotectants and lyoprotectants are used to minimize protein denaturation during freeze drying. Primary examples of protein stabilizers include polyhydric alcohols, amino acids, non-reducing sugars, and polymers and methyl cellulose. Surface active agents are widely used to minimize protein aggregation at air-water or water-solid interfaces. Antioxidants, buffers, and chelating agents are also used to stabilize proteins in solution when necessary. So friends, in this module, we have introduced ourselves to parenteral products. We discussed about the requirements of parenterals. We also learned about the factors affecting formulation of parenterals, and lastly, we had an understanding of the development of the formulation of parenterals and talked about various formula ingredients. Thank you.